So I'd like to, first of all, thank the Foundation for inviting me to come and speak with you today, and to Mark and the other organizers. So I've been charged with talking about genetics. And my topic isn't specifically about Blepharospasm because we don't know that much about the genetics of Blepharospasm. So I'm going to talk about the genetics of dystonia and also, did my mic go off? And also discuss focal dystonias, try to focus a little bit on focal dystonias in general. So there's multiple ways to classify dystonias, and one of them we've heard about already, primary versus secondary. So when there's a cause to dystonia, we call that secondary. So these can be associated with other diseases, such as Huntington's disease or Parkinson's disease, or they can be associated with drugs, trauma, stroke, infections, things like that. We call these secondary or non-primary dystonias. When the only neurologic condition is dystonia, we call this primary. And many of these primary dystonias have a hereditary predisposition, so genes are involved, and there's no consistent pathology that we know of. And so these are considered not degenerative. So unlike things like Parkinson's disease and Huntington's disease where you have degeneration in the brain, we don't see that in dystonias. And so I'm going to focus on the primary dystonias because these are the ones that we think are genetic. So another way that you can classify dystonias is based on age of onset, site of onset, and distribution. And so we've heard a little bit about this from Dr. Jankovic this morning. And this is just showing us that early onset dystonia usually occurs in the leg or the arm, so in one of the limbs, and later onset dystonia usually occurs in the neck or the face, all right? And then down here, this shows us that when you have early onset, you tend to generalize. That means you have lots of different muscles involved. And when you have later onset, you tend to remain focal, where you only have one particular body part involved, or segmental, where you have two adjacent body parts involved. And so we've heard something about the different kinds of focal dystonias. So Blepharospasm is one of the kinds of focal dystonias. There's also torticollis, dystonia of the neck, cervical dystonia. There's spasmodic dysphonia, dystonia of the larynx. And there's also writer's cramp, dystonia of the arms. So those are different focal dystonias. So another way to classify dystonias is by genetics. And so this is an ever-growing list of loci that are involved in dystonia. So there's more than 20 different loci now involved in dystonias. And the ones that I've put in green, DYT 1, 2, 4, 6, 7, 13, and 17, are what we call the primary dystonias. So this is where dystonia is the only or the main clinical symptom. Of the primary dystonias, only two of them, DYT1 and DYT6, have been identified, the genes for those. So this is TOR1A, the gene TOR1A, or Torsin-A, you've probably heard about, and then this new gene that we just found in the last few months called FAP1 for DYT6. All right. Most of the genes, the primary genes anyways, many of them are inherited as autosomal dominant traits, but they have this unique feature called reduced penetrance. So I'm going to try to explain what that is. So I thought it would be good to just go over what dominant inheritance means. So this is a pedigree. A pedigree is just a picture of a family displaying dominant inheritance. And so here, the squares are males and the circles are females. And when the circles are colored in, that means that the person is affected with whatever particular disease we're studying. And so when the other thing that we have to remember is that for any particular trait, we have two copies. So we have a copy that comes from our father and a copy that comes from our mother. All right? And so when you have a dominantly inherited disease, all you need is to have one of those traits, or alleles we call them, to be defective, and you will get the disease. You will inherit the disease. So you only need one bad copy is enough to inherit the disease, and an affected person usually has an affected parent. So you can see here's an affected child and they have an affected parent. Here's several affected children in this generation. They have an affected parent. So most often, an affected person has an affected parent. And then the other thing that we have to know about this is that because for any particular trait, there's 2 copies, this person has 1 good copy and 1 bad copy, so half the time. So each child has a 50% risk of inheriting either the good copy or the bad copy. So there's a 50% chance of being affected for each of these children. It's not 50% of your children are affected, it's that each individual child has a 50% risk. So the thing that I told you about that's unique about dystonia is this thing called reduced penetrance. So reduced penetrance means that you can have the mutation that causes the disease, but you don't have any of the clinical symptoms of that disease. And so these are actual pedigrees, families with DYT1 dystonia, and they turn out to be Ashkenazi Jewish families. And you can see that some of these families have an affected parent, the child has an affected parent, which is what we expected from what I just showed you. But many of these families, so for instance here we have 2 affected siblings, 2 brothers, and then a cousin, a first cousin over here, and there's nobody affected in between these people. But it turns out this little dot is indicating all of the people who have the mutation for this gene, but they don't have any of the clinical symptoms. And so these all are showing reduced penetrance. And so it turns out for DYT1 that the penetrance is only about 30%. And so when we think about, I'm going to go back one, yes. So when we think about dominantly inherited or recessively inherited diseases, we think what we call that they show simple inheritance or single gene disorders or Mendelian inheritance. But when we add on penetrance, that suggests that there's other factors. They can be genetic or they can be environmental that somehow interact to either increase someone's risk of developing the disease or protect them from developing the disease. So the way that we have found DYT1 and DYT6 and all the other genes that were listed on that chart that I showed you is by using this thing called positional cloning. And the way that you do positional cloning is you start with linkage analysis. And so what we start with is multi-generation families with multiple affected members, and we get a blood sample from those individuals, and we isolate the DNA from there, and then we have what we call genetic markers. So these are a series of variations that are at particular positions across all of the chromosomes. And so because we know that they're at particular positions, we then ask, do we ever see any of these markers inherited together with the disease trait? And when we see that, we call it co-segregation. That suggests that those particular markers that are showing this inheritance together with the disease are located near the disease gene. And so because we know the position of all these different markers on all the chromosomes across the whole genome, when we find one that's linked, that co-segregates, then we know the position of where the disease gene is. So we don't know the disease gene per se, but we know in the general region, out of 3 billion bases, we can come down to, maybe 1 million bases. And so then we do different techniques to try to get to the end where we actually find the disease gene. So the way that you do positional cloning and linkage analysis is you use these large families. And so this is an example from DYT6 dystonia, the gene we just found, of the family that we use to find this gene. And so you can see this is a family that's 6 generations connected, and there's more than 20 affected members in this family. So this is a huge family. This is, as a geneticist, what we like to see, but obviously it's not very nice for the family. And so the problem that we have that Dr. Fazio talked about with later onset dystonias is we don't see these kind of families. And so is the reason we don't see these kind of families because it's not genetic, or are there different reasons? And so Dr. DeFazio went over some of this data that suggests that late onset primary dystonia patients 5 to 35% of them have an affected relative. So this is called looking for familial aggregation. So just because you see something run in a family doesn't mean that it has to be genetic. So for instance, if you went to France and you looked at all the children from two parents who were French, they would all speak French. And you would say, oh, well, speaking French runs in the family. But in fact, it's because they're from France, right? So even though a trait can run in the family, it doesn't mean that it's genetic. But if you see familial aggregation of a trait, it suggests genes could be involved. So we see that some percentage of relatives of late-onset patients also have dystonias. And then we look at what we call first-degree relatives. So your first-degree relatives are your children, your brothers and sisters, and your parents. Okay, so your first-degree relatives are the ones that are most closely related to you. And we ask, what's the risk in first-degree relatives of seeing another person with focal dystonias? And it turns out that the risk in parents and in siblings is about the same. So this equal risk is consistent with this pattern of autosomal dominant inheritance, but with really reduced penetrance, so even lower penetrance than we saw in DYT1. It also suggests that this is much more complex and suggests multifactorial inheritance. So you heard multifactorial inheritance from Dr. DeFazio. So I'm going to define what that is. So you can have polygenic or you can have multifactorial. So polygenic is when you have multiple genes involved in a trait, and multifactorial is when you have genes and environment coming together to increase or decrease your risk for something. And so unlike DYT1 and DYT6, which we call simple or single gene disorders or Mendelian disorders, where that gene by itself can cause the disease, these are usually referred to as susceptibility genes, because no one gene is necessary or sufficient to cause the disease, but instead they predispose you. So different factors come together to build up and push you over a threshold that then makes you manifest the clinical symptoms. So this is just a little diagram. So we know, for instance, that DYT1 and DYT6 mutations in those genes can give you dystonia. We also know that things like certain kinds of drugs, you heard from Dr. Jankovic, certain kinds of drugs or head trauma or being in an accident or things like that can by themselves give you dystonia. But the vast majority of focal dystonias, the late onset dystonias, are going to fall in this middle part, where genes and environment are coming together and interacting and resulting in disease. So the other thing that we know about focal dystonias is that they're the majority of the dystonia cases that are out there. So the vast majority of dystonia cases are focal. And they appear singleton, which means it doesn't look like people have a family history. But for blepharospasm, 10 to 27% have an affected relative. But these relatives often go undiagnosed or are misdiagnosed unless, as Dr. DeFazio told you, they're personally examined by movement disorder specialists. So one of the reasons for that is because there's all these different kinds of focal dystonias, right? So if your doctor said to you, does anybody else in your family have what you have? You could say, oh, no, nobody else, has trouble with their eyes. But what if your doctor said, so does anybody else in your family have a little trouble when they speak? Their speech is a little difficult to understand. Or does anybody in your family complain of pain in their neck, or their neck turns to one side? Or, have you ever noticed when somebody in your family is like writing, they have this very strange posture and it looks a little bit weird? Because all of those kinds of questions would help to elicit the fact that there are other people in your family with different kinds of focal dystonias. So if the doctors don't ask the correct questions, they're not going to get the correct answers from you guys. And so that's why it's very difficult to figure out how much genetics is involved in focal dystonias, because the focal dystonias look clinically very different from each other. So another problem is that they're late onset. So because they're late onset, so the average age of onset for blepharospasm is in the mid to late 50s. So because they're late onset, many parents have already died. And so you don't know. You can't examine that parent to find out did they or did they not have that. And many children are under the age of onset. You know, you have to wait until the child gets to 45, 50 to see if they're going to manifest the disease. And so you don't get those large pedigrees with multiple affected members. And so we can't do that kind of linkage mapping that I described. So is there something else that we can do in focal dystonias to look for genes? And so the answer is we can use this alternative approach called association studies. So what's an association study? So the linkage-based study is a family study where we collect large pedigrees. So now an association study, on the other hand, is a population-based study. All right? So the vast majority of people with focal dystonia, they seem to be only a single case, but there's lots of them. So what an association study does is it looks at cases, people with blepharospasm versus controls, people who don't have blepharospasm, and it looks at the same kind of markers that we use in the linkage analysis, these variants, and it asks, is there a difference in the frequency of a particular variation in your cases versus your controls? So can we see something different about our cases versus our controls? And again, because we know the position of these different markers, if we see this difference in frequency, it suggests that something, either that marker that we're looking at itself or something around that, has something to do with the disease, because we're seeing it more frequently than we would have predicted by chance. So again, this is different from Mendelian because these are susceptibility genes, and no one gene is necessary or sufficient, but they're going to predispose. So there's two different approaches. And the standard approach was to do this candidate gene search, and I'm going to talk about that first. But recently, because of the whole Human Genome Project, I'm sure you've heard about sequencing the whole Human Genome Project and multiple things that have come from that, we have a new way to look, and that's called the genome-wide scan. So candidate gene search I'm going to talk about first, and I'm going to discuss a little bit. I'm just going to quickly go over what Dr. Fazio went over. First of all, I'm going to define what one of these kind of variations is that we use. It's called a single nucleotide polymorphism, or a SNP. And there's millions of these distributed throughout the genome. So we have 3 billion base pairs of DNA in our genome. And on average, there's a SNP every 1,000 base pairs. All right, so there's millions of these, and it's basically a single base pair substitution. So for instance, on your mom's chromosome, you could have got from your mom an A at this particular position, and from your dad you could have got a T. And so you have this variation that we can test for because it's different on your two chromosomes at this particular position. So this is a marker. So now we're going to look at DYT1. So this is the gene for early onset dystonia. So one of the reasons to look at a gene for early onset dystonia in late onset dystonia is because we know within families with early onset dystonia, there are people who only have a focal dystonia. So there's some people, although there's kids who have generalized dystonia, there's some people within the same family with the exact same mutation, and all they have is a writer's cramp, or all they have is a torticollis. So that suggests that this gene can manifest as a focal dystonia. And so there's different of these polymorphisms, these SNPs. So this is the mutation that by itself can cause early onset dystonia. But there's different variations, these SNPs that I was talking about, in different regions of the gene that we can now look at and ask, are they associated in focal dystonias, using this case control kind of study. And when this is done, it's been done many times, and you've seen this, but in a table form, which is probably easier to see than this. But it turns out that there's a polymorphism at the end of the gene that increases the risk in Iceland. And this was mainly in cervical dystonias or neck dystonias, but in Dr. Giovanni's study, DeFazio's study, it was in blepharospasm. There's no association seen in German cohort, mainly of cervical dystonia, and also in this blepharospasm cohort from the US. But then you heard that they did a reanalysis of the Italian and the US blephs, not looking at bleph itself, but looking for the spread of the disease. And when they do that, then they find that there's a consistent association in both the US and the Italian. So now, if we ask the question slightly differently, we actually get consistent results. And then there's a different SNP in the end of the gene that's been associated with protection in a German and an Austrian cohort, mostly with cervical dystonia, but with some cranial. And then there's another SNP within the gene itself that's been associated with positive family history of focal dystonia in an more isolated northern German population with cervical dystonia, blepharospasm, and writer's cramp. So you can see that we have these conflicting results where some are saying it's a risk factor and some are saying it's protection. But it suggests that all of these studies suggest that there's something within that gene. We don't understand why we're getting these differences, but there's something within that gene that's important for susceptibility to focal dystonia. So what are some of the problems with these studies? So some of the problems have been touched on already, the phenotypes. So we don't know if blepharospasm is going to, there's going to be a certain set of genes that's specific to blepharospasm and another set of genes that's specific to cervical dystonia and what have you. So we're taking, because we don't have enough of any one type, we're mixing potentially different kinds of dystonias, and is that a problem? We're also mixing focal versus segmental. So segmentals are the guys that spread. So we have people that only have a focal dystonia versus people that spread. And we also have plus and minus family history. So all of these may be confounding the heterogeneity, confounding our results. Another problem is its sample size. So the samples, we have 100 patients here, 200 patients there, at the most 300. Maybe the sample size is too small because we really have no idea how many different genes we're looking for. And then we also have isolated populations versus outbred. So we have like the Icelandic population, which is relatively isolated. We have the northern German population, which may be slightly isolated. The Italian population versus the US population. And so is there a difference in genetic factors when you have an isolated population versus an outbred population? And lastly, we're only testing a few SNPs, and we're not looking at the whole gene in general. And so maybe we have to look across the whole gene. So this gets back to my last point, which is this genome-wide association study. And so people are now using these genome-wide association studies for things like cancer and diabetes and Parkinson's disease. And so basically what this allows you to do, instead of looking at a single SNP or a couple of SNPs in a gene, is to look at SNPs across the whole genome. And so we use a chip, and we take DNA and we attach it to the chip, and we look at what we can find here, and basically we're going to get 1 million SNPs at a time on a person. All right, and so then we can ask, not just in a particular gene, but across the whole genome, do we see any associations? And of course, there's issues with this because when you're looking at 1 million of something, just by chance, you're going to find things. And so you have to sort out which are the things by chance and which are the things that are real. And so this is just a study that we've done, and we've actually focused on Ashkenazi Jews who have focal dystonia, with the idea being that when you have an isolated population like the Ashkenazi Jews, maybe the number of genetic factors contributing to their disease is smaller than when you have an outbred population. And so maybe the chances of finding those things are higher. You need less patients. And so we've only had a very small number of patients because these studies are very expensive. They're like $500 a chip to do these studies. And if you're doing 100 patients times $500, you can see how it can add up very quickly. Plus you have to do your controls. And so we have 105 patients and 400 controls. And this is called a Manhattan plot because it sort of looks like the tall buildings in Manhattan. And what you're looking for is things that come out way up here. Because by chance, anything that's down here is not significant when you correct for doing it 1 million times. And what you really want is things that are way up here. And so you can see that we have some spots way up here that are potential SNPs that are associated with focal dystonia in this population. And so now we have to figure out how to follow this up and decide if they're real or not. And so I'm going to finish with how do we actually find more genes, and the biggest thing, of course, is collaboration, because we need much more numbers. So any one center maybe has 200, 300, 400 patients, but we need thousands of dystonia patients, focal dystonia patients of all different types in order to find genes related to focal dystonia. So collaboration for families and singletons to increase the sample size and to replicate things that we find. We need more rigorous diagnosis and selection criteria. We need to make sure that all of us are, when we say that this patient has dystonia of a particular type, are talking in the same language. Because if we mix different things that aren't necessarily really primary dystonia, then that's going to mess up our results. We need to expand the families by examining the first-degree relatives and finding out, do we have any larger pedigrees that we can go back to the old strategy and use the linkage kind of analysis. And maybe we need to use ethnic isolates to reduce the number of genes in any particular isolate, find something, and then go to the outbred population and ask, does that factor also contribute to the more broad population of focal dystonia patients? And of course, the identification of more genes may help us to identify or connect functional pathways and hopefully will provide new therapeutic targets so we can get some better medicines for dystonias, focal dystonias, and dystonias in general.